Welcome to the Advanced Fluorescence Microscopy and Image Processing course. Today is the lecture number 20 and we will discuss about spatial transcriptomics and proteomics. We will discuss about the technologies such as RNAscope, MERFISH and Codex. So there are many technologies available for spatial transcriptomics and proteomics, but all we cannot cover. So I will just give you the representative three technologies. So the RNAscope and MERFISH are used for the spatial transcriptomics analysis, whereas Codex is used for spatial proteomics analysis. So let me explain you what is spatial transcriptomics or proteomics. So in one of my previous lecture, we discussed about this technique which is known as single molecule mRNA FISH to quantify the number of mRNAs per cell. Okay, so this technique is generally being used to quantify the transcription or quantify the gene expression level. And how it works, let's say if you want to quantify the transcription of this COOP1 gene, then you will make more than 25 probes, fluorescently labeled probes, which can bind to this mRNA transcript that is produced from this gene. So this is the mRNA transcript that is produced from this gene by transcription, and these fluorescently labeled probes, which are more than 25 in numbers, they will bind to this mRNA, and there are so many probes which are binding to this mRNA, so they will give a very bright fluorescent spot. Okay, so in this image you can see these are yeast cells and the nuclei are stained by DAPI staining and they are shown in green, whereas this dots, the red colored dots that you are seeing, those are the individual molecules of mRNA. So this each dot contains so many 25 probes bind to the same molecule of the mRNA. Okay, so each spot represents one mRNA molecule, and we are seeing that because of the presence of more than 25 probes which are bound to this mRNA. And you can see different cells show different expression level. Okay, let's say this cells shown by the yellow borders, so they are non-transcribing cells because there is no mRNA produced in these cells. But if you see the cells which are shown by the blue borders, so those are the actively transcribing cells, and you can see how many mRNAs have been produced per cell. Okay, so this technique we call SM FISH or the single molecule mRNA FISH, and that is used to study the number of mRNA produced per cell to quantify the transcription. Now this is at the cell level. Okay, but what will happen if we use this technique at the tissue level? Okay, so just like immunofluorescence versus immunohistochemistry. So in immunofluorescence you can do the staining of individual cells, but in the immunohistochemistry you use it for staining the tissue sample. So what will happen if you use this technique to see the mRNA expression level in the entire tissue? So here is one tissue section, and you can see different cells here, and the PDL1 gene is labeled by the SM FISH. So by making those kind of probes, here the PDL1 gene is fluorescently labeled, and here it is shown in the blue dot. So each blue dot represents one mRNA. Okay, now this is a tissue section. So what you can see in the same tissue, in this area, the expression level of this PDL1 gene is quite high because there are so many blue spots, individual mRNAs, whereas in this area, the expression level of this PDL1 gene is low. And here the expression level is medium. And if you consider this area, you can see that there is no expression. Okay, so a tissue sample can show this kind of heterogeneity in the expression of any gene. Okay, so the same gene in the same tissue, in one area it is highly expressed, in other area it is the expression level is low, and in some of the areas it is not at all expressing. Okay, so this kind of information is known as the spatial transcriptomics because you are quantifying the transcription with spatial resolution. Say how in a tissue sample, how in different areas the expression level changes. Okay, so this is known as spatial transcriptomics. Similarly, if you quantify the protein expression level in the tissue with respect to the space, then it is known as spatial proteomics. Okay, so it is possible that maybe in this area of the tissue, the protein is overexpressed. Here it is underexpressed, and here there is no expression of any particular protein. So that is known as spatial proteomics, whereas this figure is specifically for spatial transcriptomics of the PDL1 gene. So why this is important and how it is done? So spatial transcriptomics is a molecular profiling method that measures gene activity in a tissue sample and map where the activity is occurring. It examines the gene expression in the context of the tissue microenvironment. Okay, so let's say this is a tissue sample. Okay, and in different areas of this tissue, one gene has a different expression level. Okay, so using the spatial transcriptomics platforms, you can quantify that. Okay, in this area of the tissue, the protein is overexpressed, or in this area of the tissues, the protein is the gene is underexpressed. Okay, so spatial transcriptomics is equals to all mRNAs spatially resolved. Okay. So here you can see that we are quantifying the number of mRNAs per cell and they are resolved spatially. So this slide nicely depicts the differences between the bulk analysis versus single cell analysis versus single cell spatial analysis. Okay. So what is a bulk analysis? So bulk analysis means a regular mRNA-seq or the RT-PCR that you generally use for quantifying the gene expression level. So those are known as the bulk analysis. Why they are known as the bulk analysis? Because for those analysis, you take thousands of cells, you lyse them, and then from the cell lysate, you just measure the transcript levels. So what are the transcript levels you get? Those are the average transcript levels over thousands of cells. Okay, so those kind of techniques are known as the bulk measurement approaches. So those kind of bulk measurement approaches are available for transcriptomics analysis as well as proteomics analysis. Okay, but you cannot get the single cell resolution. Okay, versus single cell analysis, if you do, you can get to know that how this each cell expresses the genes. Okay, so how different genes are expressed in individual cell, that you can get to know. Okay, but what the spatial transcriptomics or the spatial proteomics provides an additional information is that how these transcripts are spatially arranged. Okay, so let's say if you see this picture, you can say that okay, in the cell or maybe in the live cells, the strawberries are arranged in these particular locations. This kiwi are arranged in these particular locations, means those transcripts are present in these particular locations. Okay, so in the spatial transcriptomics or the spatial proteomics approaches provide you the one level extra information, and that is the level of transcription or the proteins at that particular area of the tissue. Okay, so there are several platforms for spatial transcriptomics analysis. So let's say this is your tissue sample, and for getting the information about the mRNA expression level, you can either choose the NGS based platforms, that is next generation sequencing based platforms, in which the different areas of your tissue will be captured on a platform, and from each spot you can prepare the library and you can give a unique barcodes, and then you can go for the NGS, that is next generation sequencing, and then you can align your sequencing and do the data analysis, and you can reconstruct the image of a tissue, and you can get to know that in this area the gene expression was high, whereas in this area the gene expression level was low. Okay, so that is an NGS based approach. But the other two approaches are imaging based. So there are two approaches which can be done based on imaging. One is known as the in situ sequencing, and the other one is known as in situ hybridization. So in situ sequencing, the rolling amplification takes place, and then from those amplification you have to do the sequencing, and then you can align and do the data analysis to get the spatial transcriptomics. And the other method is in situ hybridization based, which is purely imaging based. Here there is no sequencing. Okay, so let's say these are the mRNA species produced in different areas of the tissue, and you can design the fluorescent probes. Those fluorescent probes will bind to those mRNAs in the tissues, and then you can image the tissue sample, and you can decode the information about the spatial mapping of gene expression. Okay, so in today's lecture, we'll mostly focus on this third type of technology, that is in situ hybridization based and imaging based technologies. We won't discuss more about the NGS based analysis and the in situ sequencing analysis. So here there is a little history about how this different platforms of spatial transcriptomics were developed. So initially this technology was started from 1982, and the first technique that was developed for spatial transcriptomics analysis was the SM fish, the single molecule mRNA fish that we just discussed. Okay, but later on, around 2008, this improved protocol of SM fish was developed, and now the one that we are discussing, which is a Stellaris probe based. So those kind of protocols were developed sometime in 2008. Then the RNAscope technology was developed around 2012, and then around 2015, MERFISH was developed. And there are so many other types of methods available for spatial transcriptomics, but we cannot discuss all of them. So in today's lecture, we'll discuss about RNAscope and MERFISH and Codex for spatial proteomics analysis. Okay, so let us start with RNAscope. So RNAscope is a method for in situ hybridization, and that is developed by the company known as Biotechne ACT. Okay, so this is again a hybridization based method. So for a regular SM fish or single molecule mRNA fish, we designed around more than 25 probes, and those 25 probes binds to the specific mRNA, and we can fluorescently visualize that mRNA. So this technique is mostly based on the same principle, but here the advancement has been done in the development of these specific probes. So these are known as the double Z probes, and now I will explain you the probe design. Okay, so why this probe look like Z shape? Okay, so here at the bottom is known as the target specific binding sites. So this region is made up of 50 base pair, and those 50 base pair are complementary to your mRNA sequence. Okay, so this Z probe binds to your target mRNA because they have 50 base pair complementarity with the mRNA sequence. Then this is known as the spacer region. Okay, and this is known as the pre-amplifier binding site. Okay, so what is the pre-amplifier binding? So if you see, so how this double Z probe works. So let's say this is your target mRNA, target sequence. So double Z probes complementary bind to this target mRNA because they have 50 base pair homology for this target sequence. Okay. Then this is a spacer region, and this area is known as the pre-amplifier binding area. So this is a pre-amplifier, and this pre-amplifier can bind to several amplifiers, and those amplifiers can bind to the fluorescently labeled molecules, labeled probes, chromogenic enzymes, or fluorophores. So these are the fluorescent dyes or the fluorescent molecules that can bind to your amplifier. So why it is required? So you can see, because of this amplifier and pre-amplifier system, so many probes can bind to this double Z probe. Okay, let's say if I fluorescently label only this double Z probe, then only 2 dye molecules can be attached to this double Z probe, right? So this concept of amplifier and pre-amplifier is to increase the number of fluorescent molecules per double Z probe. Okay, so because of this amplifier and pre-amplifier system, multiple fluorescent molecules can bind to this double Z probe that will help you visualize this mRNA. It will increase the signal. Now see, because of this thing, it increases the sensitivity. Okay, sensitivity means even only one mRNA is present in the cell, but still you can detect that mRNA because there are so many fluorescent probes will bind to that single mRNA. So because of this amplification system, the sensitivity of this assay increases. And how the specificity is increased? Why they used double Z and not the single Z probes? Okay, so by putting the double Z probe, see, so both these probes have 50 base pair homology, and this amplifier and pre-amplifier can only bind if there are 2 Z probes have bound to the target site. If only, let's say, because of the some non-specific complementarity, if only one probe bound here, then this pre-amplifier will not attach to this Z probe. Okay, so by using double Z, they increased the specificity of this assay. So this amplifier and pre-amplifier will only bind to the double Z structure, okay, not to the single Z structure. So this is how this method provides better sensitivity and better specificity compared to the regular SM fish technology. So how this technique works? So let's say on a slide, this is your tissue sample or the cell sample. Okay, mostly generally this technique is used for the tissue samples because it provides the spatial information. Okay, then you hybridize your slide with those double Z probes. So this double Z probes will bind to different RNAs. So let's say these probes are designed for RNA1. This yellow color probes are designed for RNA2. So there are multiple colors you can use, like at least 3 or 4 color simultaneous detection you can do. Okay. Then this preamplifier and amplifier and this fluorescent dyes will bind to this double Z probes to give you the fluorescent signal. Okay. So eventually in a slide, you can see the single molecules of mRNA, and each dot represents one mRNA molecule, okay, in a tissue or a cell sample. And then you can do the computational analysis. So computer can give you this kind of histograms of number of mRNAs per cell. Okay. So this is how this technique works. It is mostly same like the SM fish or the single molecule mRNA fish, but here, because of this double Z probe chemistry, the sensitivity and specificity of this assay is improved. So there are different varieties of this RNAscope technologies. You can see they are named slightly differently, and the most importantly, how you will select that which RNAscope technology you should use for your assay depending on the number of mRNAs you want to detect. So 3-plex means if you want to detect 3 mRNAs simultaneously in your tissue sample, you will go for this assay, RNAscope fluorescent multiplex. If you want to see 4 mRNAs together, you will go for RNAscope multiplex fluorescent V2. Okay. If you want to see 12 different mRNA molecules per cell or per tissue sample, then you will go for RNAscope high-plex. Okay. So based on your application, you have to select which platform or which technology you will use. Now this technique can be combined with immunofluorescence also. So why it is required? So let's say if you want to see the co-localization between some proteins and mRNA. So with the immunofluorescence using specific antibodies, you can detect your protein in the cell sample or the tissue sample, and simultaneously you can get the information about the gene expression from the same sample. Okay, so how it works? So this protocol remains more or less the same, but one step is added in which you will incubate your sample with the antibodies. So this is your slide. You take your tissue or the cell sample, then you hybridize with the double Z probes for, let's say, 3 different RNAs, red, green, and blue. And then you amplify your signal using this amplifier, pre-amplifier, and this fluorescent molecules. So all these RNAs will give you this different colored dots like red, green, and blue dots. And then you want to see your proteins. Okay, so you incubate your slide with the antibodies, primary and secondary antibodies specific for that protein, the way we do it for the immunofluorescence analysis. And then you can see your proteins labeled with the antibodies in one color, and also you can see the mRNA expression pattern. And then with the computational analysis, you can count the number of mRNAs per cell. Even you can also count that how many this orange color cells shows the mRNA expression level. Okay. So this is known as dual RNAscope, ISH-ISC, in situ hybridization, immunohistochemistry, or immunofluorescence. So here is one image of this dual ISH-IF for 3 probes. So see here in this sample, they have labeled in a tissue sample. This is a tissue sample, and they have labeled 3 mRNAs: PD1, TIM3, and PDL1. Okay, with these 3 colors. So you can see the PD1 mRNA spots are in green color. So in a tissue sample, you can see that only these cells are expressing this PD1 gene, whereas so many cells are expressing this PDL1 gene, and so many cells are expressing this TIM3 gene. Okay, and along with that, they've used CD45 immunofluorescence. So CD45 antibodies. So the white signal represents the localization of the CD45 protein. So if you see the overlaid image or the merged image, you can see these cells are expressing CD45 protein, and they are having the transcript levels for PD1, PDL1, and TIM3 as shown in this figure. From this data, you can actually quantify cell-wise the number of mRNAs for each of these genes and the number of cells that shows the CD45 marker. Okay, so this way you can combine both the technologies, ISH, that is in situ hybridization, and immunofluorescence. Now, in one of the previous slide, we observed that with this technology, you can actually detect 12 different mRNAs in the same sample, right? So how this 12 different mRNAs can be detected from the same samples? So how 12 probes can be detected from the same sample? So it is detected in the batches of 4 plus 4 plus 4. So how it works? Let's say you want to detect 12 different mRNAs. So first you hybridize your cells or the tissue with all 12 probes. Okay. So all 12 probes will hybridize to 12 different mRNAs. So all 12 different mRNAs are hybridized with this 12 different double Z probes, and they are already labeled with the pre-amplifier and the amplifiers, but the fluorescent dyes will be added in the batches of 4 plus 4 plus 4. So first only the fluorescent dye for with 4 different colors will be added for to detect these 4 mRNAs. Then you image this sample, then again wash off this sample so that these 4 fluorescent dyes will be washed off. Again you add another fluorescent probe specific for this amplifier for 4 different colors, and then you image the sample again. Then again you wash off. Again you use another 4 set of fluorescent dyes which are specific for these amplifiers. So that way, in a batches of 4 plus 4 plus 4, maximum you can go for 12 different mRNA detection in the same tissue sample. So you see, this way the different color fluorescent probes will attach to these amplifiers, and you image this sample, then wash off these probes. So these probes will go away, and then you add next batch of 4 fluorescent probes. So here is the RNAscope Hiplex assay workflow. So as I mentioned that for detecting 12 mRNAs, you need to do the same experiment in 3 rounds. Okay, so in round 1, okay, you will go for the target hybridization. So hybridization of all the 12 different genes will take place in the first step and then in the first round. Okay, so your double Z probes will bind to all 12 different mRNA species, okay, in the sample. But then while imaging, you will only add the fluorescent probe specific for target 1 to target 4 genes. Okay, and you will image that sample and capture the positions of those 4 genes in the tissue sample. Then you will wash off this tissue sample. Okay, and that is known as T1 to T4 fluorophore cleavage. So the fluorophore will be cleaved off from this target 1 to 4. Then you will add the fluorescent label for T5 to T8. Okay, target 5 to target 8. So only this target 5 to target 8 mRNAs will be fluorescently labeled. Again, you image that sample. Then again, you wash off the sample and add another. So this is the fluorophore cleavage for T5 to T8, and then you add the fluorescent probes for target 9 to target 12. Okay, and then again image the sample. So there will be 3 image for the same tissue. Okay, and those 3 image, the first image represents the positions of first 4 mRNA species, the second image represents the positions of next 4 mRNA species, and the third image represents the positions of next 4 mRNA species. And you see the time, only the first round takes 7 to 8 hours because here the hybridization step is there, and hybridization step is is same for all 12 probes. Okay, but once the hybridization is done, this adding the fluorescent dyes, imaging and washing of the fluorescent dyes, it takes just 1 hour. So the second step is only 1 hour, and the third step is only 1 hour. Okay, so basically within 10 to 12 hours you can do this sequential round of probing and imaging. So coming to the next technology, which is known as MERFISH. Okay, and that was developed by the company known as Vizgen. So MERFISH stands for multiplexed error robust fluorescence in situ hybridization, and it can detect 100 to 1000 RNA species simultaneously in a single cell using combinatorial labeling, sequential imaging, and error robust barcoding schemes. Okay, so using that RNAscope, you can detect 12 mRNAs together. But what you will do if you want to detect hundreds of or thousands of mRNAs together? So to detect so many RNA species simultaneously in the same tissue sample, you should go for MERFISH. And MERFISH method works by assigning barcodes to the cell's RNAs and hybridizing them with a library of DNA probes to present these barcodes and then reading them out by imaging to determine the identity of individual RNA molecules. So I'll explain you in the next slide how it works. So let's say in your tissue sample, there are m number of mRNAs. Okay, 1, 2, 3, 4. That way, let's say you want to detect 100 or even 1000 mRNAs. Okay. So how these probes are designed? So these are 96 into 90, means total 2 times 96 probes are there. Okay, 192 probes total, and for each mRNA. Okay, and this is the target sequence of the probe. So those target sequence will bind specifically to the mRNA, and each probe has two readout sequence. That means the two different fluorescent molecules. So this is the readout sequence 1, readout sequence 2, and similarly here this is the readout sequence 4 and readout sequence n. Okay, so these probes are known as encoding probes. Okay, so each mRNA will be labeled by 96 into 2 probes, and each probe will have two different colors of labeling, readout sequencing. Okay, so how it works? Let's say if you consider this first mRNA, so this probe will bind to this pink color dye in the first round, then it will hybridize to this blue color dye in the second round, then it will not hybridize to this green color dye because its readout sequences are not, it doesn't have the readout sequences for this green color dye. Then it will bind to this fourth dye. Okay, and then so there will be several readouts of the image. So these are readouts, means multiple images you will take after putting this fluorescent dyes. Okay, so fluorescent dyes will specifically bind to only those RNA species for which it has the readout sequence. Okay, and similarly it will give some barcode to each RNA species. So let's say for your first RNA, let's say this RNA, this is the barcode. So in first image you could see the signal, so it is number 1. Then the second image you could see the signal, so it is it is also 1. In third readout there is no signal, so here the number 0 is there, number 0 is there. In the fourth readout there is a signal, so number 1 is there. So this way each RNA will have different barcodes like 11011. Then the second RNA, you see in first 4 cycle of imaging it was not detected. See the second mRNA, there is no hybridization, no hybridization, no hybridization, no hybridization. Only the hybridization occurs in the nth round. Okay, so it has a barcode of 00001. For third mRNA, it was detected in second and third image. So in the first image, it is not there. In the second image, it was hybridized to this blue colored probe. It was hybridized to this green color probe, but it was not hybridized to this maroon color probe. Okay, so its barcode is 01100. So this way, each mRNA which will have a different or unique barcode. So here is another example to explain you that how this MERFISH work. So this is the same cell, okay, and it has, let's say, 6 different mRNAs, A, B, C, D, A, B, C, D, E, okay, and there are 2 mRNAs of the same species like A and A. So how it will be detected? So let's say in image 1, only this 4 mRNA will give fluorescence signal. So they will be given 1111 number, and those mRNA which are not giving signal in the first round, they will be given 0 number. Then in the second round, the another set of fluorescent dye will be added, and it will bind to only first and these 3 mRNA species. So those will be number like 11, then 10, because this mRNA gave signal in first round but not in the second round. So it is 10. The same thing here, 10, 01, 01, 11. In the third or the nth image, only these 2 mRNAs will give fluorescence. So they will be given number like 101, 011. Whereas these other mRNAs which are not fluorescent, they will be given number 110, 100, 110. The last digit is 0, you can see. So eventually this is the decoded image. So for this mRNA you got the barcoding 110. For this 100, this is 101, 010, 011 and 110. So these 2 barcodes are the same, 110, 110. That's why those are the same transcripts. whereas the other barcodes are different. That's why those are the other transcripts. So here is the real image of Murfish experiment. Okay, so this is a, let's say, tissue sample, and the real image of the Murfish experiment where 1001 RNA species were detected in single cell using Murfish. Okay, so real image looks like this. So each spot, those are actually artificially uh, uh, given spots. So that's why you can see them as perfect round shaped spots. Okay, so this is the reconstructed image. This is not the real image. Okay, so here you can see the magnified version of this small white box. Okay, and this small white box has so many mRNAs from starting from 1 to 12. So you can see all different mRNAs are there. Okay, total 12 mRNAs. Now you just focus on this third number mRNA. So how this third number mRNA will be barcoded? So see this table, I have highlighted this third number mRNA barcode. So this third number mRNA, it is not visible in the first image. In the second image, it is not visible. In the third image, it is visible. So it will be given number 1 here in the third image. Okay. Then again in fourth image, it is not visible. Fifth image, it is not visible. Sixth image, it is not visible. In the seventh image, it is visible. So it is given one number in the seventh. Then eighth image not present, ninth not present, 10th not present, 11th image not present. In 12th image, it is present. So again here number 1 is given. Okay, and then it is present in 14th image. So here number 1 is given. Okay, so this is the barcode for this particular mRNA 001000100000101. So this barcode, the length of this barcode is 14 digits. Okay, so using this 14 digit, and that's why it is known as 14-bit MHD2 code. Okay, so this is the barcode which is of 14 digit, and when you have to detect so many species, you need such a long barcode. But let's say you want to detect only 100 mRNAs, you may not need such a long barcode. Okay, and this transcript was identified as ZFPM2 transcript, so that is some particular gene for which this transcript is there. So in the entire tissue sample, you can or the cell sample, you can see where this transcript is present and how it is spatially expressed. Okay, so this is how, based on this unique barcoding system, this Murfish technique can detect thousands of species of mRNA in any cell or tissue sample. So now coming to this technology, which is known as Codex, and that is from Akoya Biosciences, and this technique is for spatial proteomics. So the one that we discussed till now, the RNAscope and Murfish are the technologies for spatial transcriptomics. Now this Codex is for spatial proteomics. If you want to see that how different proteins are produced with respect to the space in your tissue sample, then you will go for Codex. So Codex stands for code detection by indexing, and Codex is a method for spatially resolved multiplexed immunofluorescence, okay, protein detection. And the Codex system enables researchers to image 40 plus protein markers in tissue sample at single cell resolution. It is highly multiplexed imaging, enables the study of different cell phenotypes and how they interact in situ to impact disease pathology and progression. And as this technique is for the protein detection, obviously it is based on the antibodies, and antibodies conjugate to a proprietary library of oligonucleotides called barcodes. Okay, so let us see how this technique works. So for this technique, a special kind of antibodies are developed. Okay, so any antibody can be modified like this. So what they are doing, basically they want to attach a small oligonucleotide, means small single-stranded DNA sequence to the antibodies. So they use this maleimide modified DNA oligonucleotides. They use this chemistry so that using this maleimide, you can conjugate this antibody with a small DNA sequence, single-stranded DNA. Okay, so your antibodies will look like this. This is antibody against protein 1, this is antibody against protein 2, and this is antibody against protein 3, and 40 different antibodies or 40 different proteins you can detect using this method. And all these antibodies are, you can see, conjugated with one small barcode of DNA or the oligonucleotide or the single-stranded DNA. Okay, so that is known as Codex antibody panel. Okay, and this Codex antibody panels are available for different tissue samples, different tissue types. Okay, now what will happen? So this antibody this is a DNA conjugated antibody, okay, and it will bind to your specific protein in your tissue sample, and then you will use a complementary probe to this DNA sequence, which is fluorescently labeled, okay, and then you will image so that you can see the position of this protein in the cell or the tissue, and then you can uh, remove this probe. So chemical stripping of the fluorescent oligonucleotide. Okay, so you can also just using some chemicals, you can denature this double-stranded DNA, and the probe will be removed. Okay, so again, here is another example that how this technique works. So this is your slide. The way you prepare your immunofluorescence slide, you have to prepare this, but you have to use the Codex antibodies, which antibodies are actually labeled with a single-stranded DNA probe. So let's say here there are 1, 2, 3, 4, and 6 and 8 number antibodies. So they will bind to your different proteins. Then in the first cycle, you will fluorescently detect only first 2 antibodies. Okay. Then you take the second image, you will wash off these antibodies and or the fluorescence molecules, and then you will use the another fluorescent probes that can bind to the antibody 3 and 4. So in the second image, you will capture this data, and then you will wash off these fluorescent probes, and in the third cycle you will use the probes which will bind to this antibody 6 and 8. Okay, so in the third round these two antibodies will give fluorescence, so you will take the third image to detect these positions. So eventually you will have the multiplexed image with this way. Okay, so here is the Codex antibody panel. So for any tissue, those kind of specific panels are available from the company. You have to buy this, and then those antibodies will bind to different proteins in your tissue, and then in the you can reveal them one by one. So first you will use 3 colors to see where these 3 proteins are localized. Then again you will image them. Then you will remove these 3 fluorescent probes. Again you will add another 3 probes for another 3 proteins. Again you will go for imaging. Again you will wash off those 3 probes. So that way you can do multiple cycles to detect around 40 proteins in the same sample. So I think that's it for today, and thank you for attending today's class, and our next lecture will be on high content imaging. Thank you.